This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 359, recorded on October 14th, 2015. Hey everybody, I'm Vincent Racaniello, and you are listening to TWiV, the podcast all about viruses. I have a special episode for you today. Visiting me here in the TWiV studio from the University of North Carolina in Chapel Hill. She's a professor in the Department of Microbiology and Immunology, Blossom Damania. Welcome.
Hi, everyone.
And not only is she here, and we're going to talk about her work, but this is the first ever TWiV in front of the wall of polio.
Yes.
How
awesome.
about that? Because I didn't have this in here previously for all my other in-studio recordings.
I'm so privileged.
Isn't that cool? You can see the little sign here. And you actually made a new sign for me, right?
Exactly, and you can pin that up.
So you're here to do a seminar, right? And you've been visiting. You have a good visit?
Oh, awesome visit. Yeah, I enjoyed talking to everybody. I was running late everywhere I went, so
Good.
I apologize for that.
Even though there's only 3 virologists here, it's still fun, right? Which I complained to you last night on Twitter. So what I'd like to do is talk a little bit about your upbringing and education and how you got into science and where you are today. And then let's talk about your virus and the cool things that you're doing with it, okay?
Okay, sounds good.
So you were born in India.
Yes.
Where?
I was born in Bombay, also known as Mumbai.
But you can't use that word.
I can. It's very hard for me to say Mumbai when I've been saying Bombay all my life. So,
right.
So I was born there, and my family moved around a bit. And so my family's lived in Switzerland, France, England. But I moved here when I was 18 to go to undergraduate college at Mount Holyoke.
Right.
So, and I've been here ever since, and I don't want to leave.
So when you went to Mount Holyoke, was it to be a science major?
Yes, I knew I was interested in biology. I didn't know what kind of biology. I just knew I was interested in biology. And I ended up majoring in biochemistry. And I had an awesome biochemistry professor who got me thinking about doing research as a career.
It's usually a catalyst, some kind of teacher somewhere, right?
Yes.
Can you do me a favor and move the hair there back?
Okay.
Because it's hitting the mic, and I know from experience that the sound of hair
yes.
hitting the mic is...
Okay.
And it may creep back, in which case I'll tell you.
Yeah.
Were your parents scientists?
No. So my dad was a businessman and worked for a company.
That's why you traveled a lot?
That's why we travel. He got stationed at different places. And my mom is a very artsy person. She makes dresses, and she wanted me to follow in her footsteps. So neither of my parents were scientists, and they were wondering how I, got the genes I got, because I was always very interested in math and science as a young kid, and then.
So at Holyoke, you said a biochem professor really turned you on.
Yes, into research, right.
Did you do some research there?
Yes. So I did biochemical kinds of research, working on enzymatic functions and doing KM plots and things like that. So Michaelis-Menten was figured hugely in my research at.
So you never wanted to go to medical school?
No. So I thought that was the only option until I encountered my professor, who had been a researcher. She worked on bacteria, and so she got me really interested in the concept of you can do research as a career.
It's interesting. Did you happen to hear the John Stites TWiV?
Yes.
She was going to be a doctor. She got into medical school, and she saw the doctors were all men. So she went into science instead. That was really good.
Yeah.
So at the end of Holyoke, you had clearly decided to go for a PhD.
Yes.
And
yes.
Is that something your research professors suggested that would be good to do?
Yes. So she had instilled that idea in my mind, and the more I thought about it, the more I liked it. And so I thought, okay, I want to do research. I want to get a PhD. And so I ended up at University of Pennsylvania for graduate school. And when I joined the program, it was one of these umbrella programs, so you have many different subjects. And
right.
And I actually liked that because I wasn't sure what I wanted to do, so I didn't know much about viruses at the time, and what I thought I wanted to do is do more cancer biology. So understand how normal cells turn into cancer cells, what changes, what pathways change. So that's what I thought I wanted to do. So I did 2 rotations in cancer cell biology type labs, and then my 3rd rotation was in Jim Allwine's lab, and he actually studied the intersection of viruses and cancer. He worked on SV40. And so that got me hooked, because here, like, I had the cancer aspect of the research, but then I fell in love with viruses and all the neat little tricks they use to manipulate the cell and make it do what they want to do and not what the cell wants to do. And so that was very exciting.
How was it to be in Jim Allwine's lab? He's a nice guy.
He's a really super guy, and he's one of those advisors that say, well, find yourself a project, right? So he didn't package a little project for me. He just asked me to find myself a project working on SV40 large T antigen. And so I remember this very clearly, because now as a mentor myself, I can see this in other students where I came in, I didn't have a lot of experience, because I had been working on enzymes and doing, very in vitro assays. So the first couple of years, nothing worked, right? So everything I tried didn't work. But he was so patient, and he just said, well, try this and try that. And so I did, and then suddenly things started to work. And then when they started working, other projects started working. And so it was a very interesting time, because I kept doubting myself for the first couple of years, wondering, did I make the right choice? Am I going to be good at this? But he just encouraged me. He was very patient. He never said, oh, you're wasting my money, or or something like that. And so, and then it paid off, because everything else after that point, there was a turning point at the end of my 2nd year where my project started to take off. And then I had 3 projects take off. I had 3 first author papers from his lab. And I haven't looked back since. But it was because he was so patient with me.
So it's so important to have a good mentor, right?
Yes.
Because you can imagine someone who is less involved and just let you go, and you may never have reached that turning point,
yes,
right? Which is key for your career, because then everything follows from it.
Yes.
I know the Jim, because I saw him at many meetings, and I remember at Gordon conferences, he would have a poster, even late in his career, and he always had a bottle of sherry to attract people to it. They sound like Jim, right?
Yes.
And you'd come up and he'd say, here, and then you'd stand in front and he would
Exactly. That sounds like Jim. And he also had bottles of scotch in his office. So
I should do a podcast with him, right?
Yes, you should.
Wouldn't he have like a wealth of cool things?
Yeah, and lots of good stories. And
do you have any good Jim Allwine stories?
There's sort of folklore that was in the lab when I joined. Like, he had a temper, but by the time I got there, he was much tamer and a kinder, gentler person. So there were stories that would fly around the lab and stuff like that. But no, he was great, and I couldn't have asked for a better mentor.
So tell us about your postdoc.
And so then for my postdoc, so I was working on SV40 large D antigen, very basic transcription kinds of stuff. So
kind of stuff we can't do anymore.
Exactly. Not in this current funding.
Not with SV40. You can do it with KSHV, right?
Yes.
You can ask what transcripts you need for something.
Yes.
We have great tools now, right?
Yes.
Can you imagine back then the profiling that we can do if you had had it?
I know. It'd be great. And it's funny because, SV40 is a polyoma virus, of course, and back then there was JCV and BKV that were thought to be transforming in humans and associated with brain cancer. But then, 10 years after I graduated, Yuan Chang and Patrick Moore, who discovered KSHV as well, found Merkel cell polyomavirus, another polyomavirus associated with cancer. So I think to myself, I could have stayed in the polyomavirus field, right? But what got me excited as I was about to graduate was the fact that Yuan Chang and Pat Moore, who were at Columbia, discovered KSHV about 3 years before I graduated. And so here was this new virus. It had a ton of open reading frames compared to my little small polyomavirus, and there was so much unknown. And so when I interviewed with Ron, he had just isolated Rhesus rhadinovirus, which is the Rhesus homologue of KSHV. And so here, a whole new virus as well. So there was KSHV and then there was RRV. And so there was so much unexplored territory. And I thought, oh, this is ideal for a postdoc. And I asked him the questions, if I do find stuff, can I take it to my own lab when I leave? And he said, sure, no problem.
And this was at the New England
Primate Center at Harvard Medical School, yes.
So this is kind of a little bit outside of Boston.
Exactly.
Right? Which they've now closed,
Yes.
that probably. And he's gone to Florida, right?
I know. He's gone to warmer weather, shall we say. But yes. So, and so we were part of the Department of Microbiology.
Do you ever go into the medical school and go to seminars or anything?
Yes, and the postdocs had to present at the departmental seminar series. We went to departmental retreats and things like that. And Ron also had projects with people in the medical school, like Dave Knipe. They had a big vaccine project together using herpesviruses as vectors.
Right.
So we interacted with the medical school quite a lot.
I went to this virology retreat a couple of years ago. I was invited.
Really? Oh, wow.
As a keynote speaker, they have an outside person.
Yes.
And they wanted me to do a podcast.
Great, yeah.
So I did one with David Knipe.
Oh, great.
And Priscilla
Yang, yes.
right? She's a relatively new faculty.
Yes, working on HCV.
And a student, Phil Kranzush.
Okay.
And in front of the audience, it was fun. They liked it.
And this was at Cape Cod? Or?
It was somewhere north of Boston.
Okay.
So
and they had lobster dinner?
Exeter, New Hampshire. No, I don't remember. So we drove along the coast for an hour, and it was in a little town and in a small hotel.
Okay, yeah.
Right? And many people in the department were there, not everybody, but all the students were there, and they made virus cookies one day. It was fun. Is that a tradition?
Yeah.
It was funny. It was really good. I had a good time.
It's like Geeks United.
I like it when people say, we want you to be our keynote, but do a podcast. So that's really looking different.
Yes.
Looking outside of the box, which I think is really, and I always enjoy doing that.
Yeah.
So I like it. Good atmosphere there. So you worked outside there in the Primate Center on KSHV, right?
Right, and also RRV. So and KSHV. Jay Jung, who used to be Ron's postdoc but had an independent position at the time, was right next door. And so, to be honest, I saw Jay much more often than Ron, because Ron was always traveling and going to meetings and things like that. And so I learned a lot from Jay. And he was always around to ask questions. And if I had some experiments that didn't work, he was ever ready to talk about troubleshooting. And so I learned so much from him as well. So having both Ron and Jay were great.
So how long did you spend there?
If you ask Ron,
Too long?
I spent too little time.
Too little.
I was there a year and a half, and I had 2 first author papers, and I asked Ron, do you think it's time for me to start applying for jobs? And he said, oh, but you're so young. And he said, well, I guess it can't hurt. But I don't think he ever thought that I would actually get a job, because back then people did postdocs for 3 to 5 years. And so even now they do that. And so he said, sure, I'll support you. And within a week of that conversation, UNC had this open search. And Nancy Rabtrub, who was chair of that search, sent the letter to Ron saying, if you have any postdocs that are interested in applying, please let them know. And so he forwarded me that email. And he said, this is a great place. I've been there. I love all the virologists there. You should apply. And so I did. And so the rest is pretty much history. And I went down, I interviewed, and
so that's the only job you ever looked at?
Yes, pretty much.
Wow, that's great.
Yes.
People look at tons of jobs
I know,
all over the place.
Yes.
That was good. You saved time.
Yes, and just because the search opened right as we had that conversation, so I didn't even start to look at many places. I think maybe applied to 2 more in addition, but everything moved so fast with UNC that.
So this is a great department, lots of good virology, cancer biology as well. One of your colleagues is coming here in January to give a talk, Stan Lemon.
Yes,
I'm his host, and hopefully I'll do the same thing with him, right? I take every opportunity. But as you know, we don't have many virologists coming through here because there aren't so many of us to host. So you took with you from de Rosier's lab, KSHV, and RRV,
right.
Rhesus rhadinovirus. Who had isolated that, Ron?
Yes. yes.
He had a colony that he was looking at, and
Right.
and they
And
just
they-
found one of his monkeys in his colony. Right?
Yeah, so based
Wow.
on the KSHV sequence,
I
they generated primers and amplified.
But they had no reason to think the monkeys were sick in any way. They just
No,
looked, right?
yeah.
So did multiple monkeys have the
Yes,
virus?
most of the colony was positive. And so most of these herpes viruses exist in normal, healthy individuals and don't really cause disease, but they're there. And so they just screen the healthy colony.
So is this a colony that was captured or bred somewhere?
They bred them at
So
the
somehow
primate
they
center.
get infected in the original
Yes.
breeding colony, right?
Right,
So there are
right.
other monkeys that are infected. It's probably been going on for years.
Yes.
From the first monkeys that were captive, right?
Exactly, yes, yeah.
Because they don't derive them from plasmids, you know?
Right. No, they don't.
Like they could do with viruses.
And so and so it was it was mother to child transmissions, so in order to get RV negative monkeys, you had to hand rear the babies away from their mothers, so that they wouldn't get RV. So you could actually then infect with RV.
It's transmitted in saliva.
Yes. Yes.
And the monkeys never get sick from this.
So they will get sick in the context of SIV co-infection.
Oh, so if you
Yes.
give them SIV,
Yes.
then the RRV replicates lytically?
It replicates lytically, but it's also latent. And Scott Wong, who is at the Oregon Primate Center, has a really nice paper showing that when you co-infect SIV and RRV animals, they have lymphoproliferative diseases, so they develop lymphoma.
And this virus RRV grows well in cell culture.
Yes,
so
that
you like to work with.
Yes, the things that you can actually get 10 to the 6 PFU, which for herpes virus is amazing,
And
except simplex, which replicates very well, but most of the
and
others.
KSHV doesn't replicate well.
No, it doesn't replicate well, and we don't have a plaque assay, so we do genomes per mil when we quantitate the virus.
Okay, so let's talk a little bit about KSHV and biology. It's a herpes virus, a gamma herpes
Yes,
virus, right?
yes, yes.
And the other gamma is EBV.
Yes, a
Which,
human.
see, I listened to your seminar.
I'm glad that.
Big DNA genomes.
Yes.
Right? And lots of open reading frames.
Yes.
And most of them we don't know what they encode, right?
A lot of them, yes, yes.
And also you said lots of microRNAs and
yes.
long non-coding.
Exactly, yes.
So there's
This genome is exploding with things that it's making that we have no idea what it's doing.
So KSHV was discovered because of the AIDS epidemic.
Exactly.
Right? But we knew that the Kaposi sarcoma was around for, what, 100 years before that?
Yes. So in 1872 is when Maurice Kaposi described the cancer.
It was in Europe, right?
Yes. He was a Hungarian dermatologist.
Mm-hmm.
He worked in the Austrian clinic, so.
So this cancer is most apparent as these blotches on the skin,
Yes.
right?
Yes, and lower legs, yeah.
And there were a certain cohort in the Mediterranean area who used to develop this, right?
Yes.
Mostly older men around the Mediterranean basin.
Exactly, yeah.
And knowing what we know now, they were probably immunosuppressed.
Yes,
right?
as you age, most people's immune system does get a little bit immunosuppressed, but they weren't overtly immunosuppressed.
They didn't have AIDS, though, right?
And they did not have AIDS. They did not have any overt immunosuppression, but they were aging. So
if you went for an organ transplant and you get immunosuppressed, could you develop KS?
yes.
And
So
that's been documented.
yes.
Yeah.
So KS is a problem in renal transplants recipients.
Okay. So then with AIDS in the 80s, all of a sudden people showing up with Kaposi sarcoma,
Yes.
which had previously been very rare.
Exactly.
Right?
And there's this explosion of Kaposi sarcoma in the U.S. and in Europe. People actually back then didn't have a diagnostic for HIV. So if a young man in his 20s or 30s showed up at the clinic with Kaposi, they would ask him, do you know your HIV status? And lo and behold, they would be positive. So it was actually a diagnostic marker for HIV way back when.
And then Chang and Moore here at Columbia identified virus, which they called KSHV, or HHV8, right?
Yeah, so they called it KSHV, but then some other folks wanted it to be called HHV. Nomenclature with virology, oh my God, it causes all kinds of discussions and arguments. So I've stuck with the KSH because they discovered it, they're allowed to call it what they want to call it. And I mean, they're just amazing because they found 2 viruses now and have linked it to disease. Because it's easy to find viruses these days with deep sequencing, but linking it to a disease
very tough,
and then other people confirming that, yes, you're right, is very hard. And so,
That's pretty remarkable, too, yeah. I'm going to visit Pittsburgh in January.
Oh, awesome. Yeah, so you'll see them there.
So then with the virus, then we could start asking how many people have infection, right? And
Yes.
so what percentage of the population are infected with KSHV?
Yeah, so it's very geographically dependent. So in Africa, close to 100%. In some areas of sub-Saharan Africa are KSHV positive. In Italy and other areas around the Mediterranean, it's more like between 40 to 60%. In the US, it's less than 10%.
And we think it's transmitted shortly after birth from the mother, is that right?
Yes, so it can be transmitted through things like breast milk, but it can also be transmitted by saliva as well as bodily fluids. Because initially it was thought that it only occurred in men who have sex with men or MSMs. But then clearly from Africa, we've realized that it's not just MSMs. You see young children who are 6 years of age with KS. So.
But it can be transmitted sexually, right?
Yes, yes. So it is an STD, yes.
And so, of all the people that have it, what did you say in the U.S., 10%?
Yeah, less than 10% is thought.
So what are the diseases that the virus is associated with?
So Kaposi sarcoma is the most common disease. But
and by the way,
yes,
it's not just on your skin.
Right, yeah. And
so you said today that you have lesions in internal organs, right?
Exactly, yes. And that's usually, so the lesions on the skin are usually treated ectopically. But what you don't realize are the lesions within, so in organs. And so a lot of the disseminated KS that kills people with HIV is due to internal KS, more so.
So if you see someone with lesions, they probably also have internal ones as well, and that's what kills you, right?
Yeah, yeah.
Okay.
And so, so it's associated with KS, but it's also associated with two different B lymphoproliferative diseases called primary fusion lymphoma, which is a B-cell lymphoma, and multicentric Castleman's disease, which is also a type of B lymphoproliferation.
So the virus targets B cells?
Yes.
And endothelial,
endothelial cells are the
and that's what causes the tumors?
Yes.
Now, for someone who's seropositive, say, to develop one of these tumors or cancers, they would most likely be immunosuppressed.
Yes.
Either AIDS or
transplant patient.
Get immunosuppressed, or measles virus infection would immunosuppress you as well. It's known to be really immunosuppressed. I don't know if it's associated with KS. Okay. And of the 10%, say in the U.S. who are positive, how many of those would go on to develop a cancer of some kind?
Yeah. So I think unless they are immunosuppressed, as you mentioned, I think very few would develop cancer in the absence of those instances. So it's very rare. In general, most viral cancers are pretty rare, and usually they occur only in the context of some sort of immunosuppression. Doesn't have to be HIV, but...
Okay. Yes.
And also,
it's one of very few viruses that cause human cancers, right?
Right, yeah.
KS, you got
Epstein-Barr.
Yes. HIV is also associated.
Yeah, so HIV is thought to be associated because of the immune suppression, and that allows other viral, tumor viruses to...
Hep C,
yes.
Hep B,
yes.
The polyomavirus,
Merkel cell. Yes.
What am I missing? I'm sure I'm missing some.
Human papillomavirus.
HPVs, of course.
Yes.
For which we can prevent them by immunization.
Yes.
So please immunize your kids.
Yes, both girls and boys.
Yeah, for sure.
Yeah.
We talk about that a lot on TWiV. 7? I thought there were more. I thought there were 11. But did we miss any?
HTLV is...
yes, HTLV.
Yeah.
Right. And there may be more that we don't know about.
Right. Yeah.
Okay, so some of the things that you've been interested in are how the virus transforms cells,
yes.
right?
Yes.
And also today you talked about innate immunity and innate evasion. Could we touch on both?
Well, I was speaking in the Department of Immunology. So.
That's right. So let's talk a little bit about lytic latent. So this is like a typical herpes virus where it can infect cells lytically, where it replicates and kills them and produces virus particles.
Right.
Or the genome can be silent, right?
Yes.
So you said some things about the latent stage of KSHV that are interesting. The state of the genome and the nucleus.
Yes.
Tell us about that.
Yeah. So the virus exists in a latent stage, and latency is the default for KSHV. So once the virus infects a cell, usually it will go latent after a couple of rounds of lytic replication. And in the body too, it's known that latency is the default pathway. And during latency, the virus is injected into the nucleus by the viral capsids. And the DNA then forms an episome, which is like a gigantic plasmid. And there's a protein called KSHV-LANA, which can tether the viral genome or viral episome to host chromosomes. So what it does is that one arm of LANA binds the viral genome at the terminal repeat region, and then the other arm of LANA binds to histones that dot your chromosomes. And so by doing that, it tethers the viral genome to the host chromosome. And in a typically latently infected B cell, there are about 40 to 50 copies of the KSHV episome in the nucleus.
So every chromosome could, in theory, have one, but
yes, yeah.
there are probably some that have multiple ones.
Right, and nobody's shown if there's a preference for which chromosome is tethered to the viral genome. I think it's pretty random.
So in this tethered form,
yes,
it segregates with the chromosomes, right? So,
yes.
And you said it's copied by cellular
Yes,
call?
yeah. So it uses host DNA polymerase to replicate its episome. So as the host DNA polymerase is replicating the chromosome, it just hops onto the viral genome, replicates it, and then keeps going down the host chromosome, hops onto another viral genome and replicates it. And so each of the daughter cells get equal partitioning of the viral episomes. So,
and this is because the viral DNA polymerase is not made.
No,
right?
not during latency. Yeah.
So are all genes off during latency?
No, there are a handful of genes that are on. So there are a handful of proteins that we know are made, including LANA, which is the protein that tethers.
So that always has to be made.
That has to be made, yeah. Without that, you would not have latency. And so
So if you take that away from the virus, it doesn't go late. It just stays in the lytic phase?
Right. So some people have knocked down LANA and shown that you cannot establish latency. So LANA is actually a hot protein to target to,
Isn't Lana a singer? Lana what? What's her name?
Well, there's an actress named Lana Turner.
There's
but
Lana Turner, but there's a singer. Now I'm gonna. What does it stand for?
A latency-associated nuclear antigen.
Okay. So this is different from herpes simplex, where just a lat RNA is produced, nothing else, right?
Yes, and I have to say that in addition to the proteins, all the viral microRNAs seem to be expressed during latency, as well as several long non-coding RNAs. So
How many would that be?
viral RNAs between 12, 14, depending on how you classify them.
And we don't know what they're doing, right?
Yeah, so there's some labs have shown that they target different things, but exactly in terms of latency, what they're doing to help latency, we're not exactly sure. Some of them degrade RTA, which is the major lytic switch protein, so preventing lytic replication.
You don't work on those
I do not work on the microRNAs, although I was on the paper that first described them for KSHV, which was Brian Cullen, was the corresponding author of that paper. MicroRNAs had just been discovered, and he's just down the road at Duke from UNC, and so he called me up and said, can we get all of your cell lines?
Everything. It's okay.
Yes. So
and you're okay with that?
I was fine with that, and we had a collaboration.
Great. Wonderful.
We actually contributed a figure to the paper.
So presumably, the idea is that these microRNAs are somehow involved in latency, but
Yes.
exactly how, you don't know. Okay.
Yeah.
So then what triggers the switch to lytic replication? So in lytic replication, the viral genome would be expressed, and you'd make viral proteins and
Yes.
virions, right?
Yes.
So what triggers that?
Yeah, so there's lots of different theories. Our lab has shown that, at least in latently infected B cells, if we infect or activate TLR7 and 8, we can get virus to reactivate and replicate. So we published that back in 2008 or 9, showing that secondary viral infection can trigger KSHV to reactivate and replicate. Other labs have looked at other like hormones and cytokines that induce reactivation. And so, but in nature,
does light or stress or UV?
Yeah, exactly. Stress, it reactivates all herpes viruses.
Right.
So yeah, I mean, if you run around with a cold sore, people ask you if you're stressed. So it's it's always been linked in some form to virus reactivation.
So, I guess HIV would also activate it.
Yes.
So do you need lytic cycle to make a tumor, or is latent sufficient?
That's a great question. So most of the tumor cell contain latent cells, but a fraction of the tumor cell is lytic. And what's thought is that it might not be undergoing complete lytic replication, but abortive lytic replication, because a lot of the lytic genes that come on early in the lytic cycle induce angiogenic factors, pro-growth factors. And so they're thought to be this paracrine mechanism where this small amount of lytic expression induces all these cell growth factors that help the tumor to grow. And so, but all the tumor cells are mostly latent, except for these, like between 2 to 5% of lytic.
Okay, and they're producing things that will then, the growth factors in the other cells around them, so that causes the tumor to grow. So why does the virus want to keep the cells dividing? If it's in a latent state, it shouldn't matter, should it? Or is it just to keep the DNA? Otherwise, the cells would die and the DNA would be
exactly.
gone.
Yes. Yeah. So it's a form of virus survival. And actually, Kaposi sarcoma is one of the most highly angiogenic cancers in the human population. And if you look at the lesions, they're reddish purple, and that's because the virus induces this whopping amount of vascular endothelial growth factor called VEGF, which is key to recruiting blood vessels to the site of the infection. So, that helps the tumor grow.
Is there a viral gene product that induces VEGF?
Yes, yeah. So there are several of them actually. And so viral GPCR, which has been shown by several labs. And
so the virus, lytic replication induces growth factor production, so the cells divide, and then eventually they accumulate other mutations, which makes them cancer cells.
Exactly.
Actually.
Yes.
Just growing is not enough, right?
Yes. Exactly.
Are there specific? Probably not, but are there specific mutations that accumulate in Kaposi's induced cancer?
Yeah, nobody has found those yet. Cancer is a multi-step pathway, so we think that the virus contributes 1 or 2 steps in that pathway, but then additional mutations which accumulate also contribute. And so nobody's found that in Kaposi's, but in lymphoma, people have shown additional mutations in the host genome that may also be causative.
So is the transformation process part of the interest in your lab? You said it's about half your lab, right?
Yes, yes. So I started, so as I mentioned, when I went into grad school, I was interested in cancer biology. So I've always had that interest in the background, even though I started working on viruses. So when I first started my lab, I wanted to work on the intersection of viruses and cancer. And I was actually hired by the Cancer Center at UNC. So we started looking at this viral protein called K1 that activates signal transduction pathways in endothelial cells and B cells. And we found that this viral protein potently activated PI3 kinase signaling. And so we ended up studying how the virus does that. It interacts with P85, which is a subunit of PI3 kinase, and then downstream activates AKT kinase and mTOR and impacts cell survival. So this protein helps the cell to survive against a lot of apoptotic stimuli. And so we started doing that, and then looking at other proteins as well. We study a viral IL-6 homolog. So this virus has a homolog of human IL-6, so very cool virus, because it has all these little pirated genes from the host genome that they resemble the host gene in the sense that you can find them by a blast search, but they've sort of either augmented the function of the host gene or subverted it.
In the case of the IL-6, what does it do?
So it augments the function. So usually human IL-6 requires a quaternary complex of GP130 and IL-6 receptor alpha to function. So human IL-6 has to bind to this quaternary complex on this plasma membrane to activate the signal transduction network in the cell. The viral IL-6 doesn't need IL-6 receptor alpha, just needs GP130. So just with 2 proteins, it can activate the same pathway.
And what's the end result of the activation?
So it activates, so it's one of the viral proteins that activates things like VEGF and STAT3 is highly activated. And these are all known to be pro-oncogenic kinds of factors.
Okay. So does the monkey RRV ever transform cells?
So the in vitro, you mean?
Or in monkeys?
Scott Wong's group has shown that the RRV infected animals do get lymphomas in the context of
another
SIV.
virus infection. Yeah, SIV. Right?
Yes,
okay.
SIV.
You may have said that.
Yeah. So
it's probably a similar thing, that it's inducing growth factor proliferation and so forth.
Right. And I didn't mention this, but human KSHV doesn't infect mice. And so it's hard to have an animal model system, which is why people study RRV. Jay Zhang's group has shown that KSHV-infected marmosets actually develop KS, Kaposi sarcoma. But again, these are primate models, so
it's hard,
yeah, it's hard.
But RRV will not infect mice, right?
Oh, I'm not sure anybody's ever tried that.
It would be easier than a primate,
yes.
right?
Yes, and less expensive. Yeah.
Yeah, because even though marmosets are a little better,
Yeah.
they're still it's hard.
Yeah.
It's still expensive and hard to do.
Yeah.
So you can use RRV somewhat as a model for studying things you can't address otherwise. And so someone asked you about a knockout mouse, and you can't do that with KSHV,
Yes.
right?
How I'd love to do that, but yes, I mean, especially when you write papers and grants and people ask you to do those experiments, and you're like, ah.
It's funny, when you write a grant, they say you should have a mouse model. Then if you have a mouse model, they say, what's the relevance of this model, so you can't win, right?
Yeah. Yeah, you really can't win with reviewers.
It's so weird, and no matter what you do, it's always, so you stay away from the animals whatsoever. So if RRV replicated in mice, that would be good. You should check it out.
Yeah, we should try that and see.
I mean, you could just check mouse cells initially to see if they're susceptible and permissive.
Yeah, in vitro we can infect mouse cells, no problem, even with KSHV, but
not mice. Yeah,
but not mice. Yeah.
How about interferon knockout mice? Maybe that's the thing.
Yes, so I believe Jay Zhang tried that a long time ago, and it didn't work.
Because that was the key to polio. But polio transgenics, the virus only replicates in the spinal cord, in neurons.
Okay.
But the receptor is in many places that we've put in as a transgene. If you cross that with an IFNAR, an interferon type 1 receptor knockout, then the virus replicates wherever there's a receptor.
Oh, wow. Oh, okay.
Yeah, so anyway, try it.
Yeah.
So then the other thing you talked about, which is really interesting, is sensing.
Yes.
Can we talk about that?
Yes.
Let's, first of all, when the virus, last week I did a TWiV with Ileana Christea.
Yes.
Who works on sensing of herpes viruses, right? The virus comes in and it has a tegument. Actually, let's look at your keychain that you gave me, right? Because it's got it right here.
Yep.
This is the herpes virus keychain, right?
You'll probably have to hold it up.
Yeah, I could stick it in later. But this is a keychain with this is a herpes virus particle.
Exactly.
And it opens it up, and you can see that's the capsid inside, which is a icosahedral capsid, and this is a lipid membrane.
Right. And the area in between the capsid and the lipid membrane is air, but that's the tegument.
This is the coolest thing.
Yes, it's cool, isn't it? I love it.
That's got to be my keychain. This has got to be with my virus license plate, a virus keychain.
And they're DNA viruses, because you're such an RNA virologist. So
that's what I work on.
Yes.
But, we're doing a DNA virus podcast here. We do probably more RNA virus TWiVs than DNA, right?
Yes.
But I try and do them when we can. So the tegument is full of proteins and RNA, you said?
Yes, and also RNA, viral microRNAs, as well as viral transcripts.
Is there DNA, viral DNA in there too?
Nobody has found DNA in there.
It's in the capsid itself, right?
Yes, yeah.
So you said today when this membrane fuses and the tegument comes out, those RNAs are sensed.
Yes, and so that's what we think is happening.
Because I'd never heard that before. You didn't publish this, right?
No, and it's hard to show that because you would have to suck out the RNA from the tegument, which would then render the virus inactive. So, yeah.
All you know is that when it infects, there's some signaling through RNA sensors, right?
Yes, exactly. And so we think that viral microRNAs, which are not capped, or long non-coding RNAs, which are not capped from the virus, could potentially trigger things like RIG-I, which recognize the 5' ends of RNA. And so, so we think that that's why we're seeing a response. Also, the herpes virus genome now with all this high throughput sequencing, it looks like it's being transcribed everywhere, right? And there are all these like double-stranded regions, they're antisense transcripts. So you can imagine a lot of secondary structure that could potentially bind RIG-I.
So you can't make a virus without any RNAs in the tegument.
That's the problem, right?
Right. But you did show that if you knock out the sensor pathways that are known to be just RNA, that you get better virus replication.
Exactly.
Right? And then someone asked you, well, I think it's RIG-I you looked at, right?
Yes.
Doesn't RIG-I recognize DNA? And you said, I don't know. I never heard of that. That would be bad for, right?
Yes.
But in the cytoplasm, that shouldn't be happening. There shouldn't be any viral DNA.
Yeah, so
right?
I don't think RIG-I recognizes DNA. I think he was talking about this paper from Veet Hornung's lab, which showed that DNA can get transcribed by RNA polymerase III into these little RNAs that are not capped, that have 5' triphosphates, and then that binds RIG-I. So I think that's what he was talking about, but he asked me if RIG-I bound DNA, and I was pretty sure it didn't.
So that's the key here, that these RNAs that are packaged are not capped, so they have triphosphates, which are RIG-I. That's what RIG-I looks like, right?
Yes, and also double-stranded RNA.
And you found also double-stranded RNAs?
Yeah,
in the
when we did
right?
immunofluorescence with an anti-double-stranded RNA antibody.
That's very cool. So this must, or maybe for the other herpes, they all have teguments, right?
Yes, they all have teguments, and they all have RNAs and protein packaged in there.
So when are you going to publish this soon?
Yes, I hope to.
It looks like it's almost ready. It's a lot of data, right?
Yeah, well, we're doing RIG-I IPs. I want to know which part of the viral genome the RNA transcript is bound
I'm sure
to.
everyone will look at their favorite herpes virus and see if this is a good.
Yeah.
So you would view this as a way, because herpes viruses are unique, that they package these kinds of RNAs.
Yes.
It's a way to sense it before the DNA even gets into the nucleus, right?
Yes. And I have a hypothesis as to why the viruses are doing this.
Putting RNAs in there?
Yeah, I think the virus actually wants to activate innate immunity to a low degree. Like, I don't think it wants to activate innate immunity to a huge amount. But I think the low level of innate immunity helps the virus then establish latency. Because without the innate immunity, it would be rampantly lytic. And it would be hard to establish the latent state. I think the virus sort of senses that, okay, there's too much interferon around, I better go latent. And so I know I'm personalizing the virus, but I think that's my hypothesis as to why it's activating interferon to a certain degree.
But you don't think these RNAs are there just for that purpose?
No,
right?
and
some other...
right. I think that these RNAs, a lot of them are viral transcripts, so they can start getting translated right away. They don't have to wait for the virus to go into the nucleus and get transcribed. The microRNAs may have targets in the cytoplasmic host RNAs, and
so some of them are capped, right?
Yes, and some of them are capped.
So yes. So with herpes, one of the proteins in the tegument, I think it's VHS. It's needed for immediate early gene transcription, right?
Yes.
Is there a similar situation for KSHV?
No, so we don't have a VHS in the tegument like herpes simplex. So our major lytic switch protein is RTA, which has to get transcribed inside the nucleus first.
Okay.
Yeah.
But incoming virions, these RNAs may have some role in early transcription, maybe?
Right. Yeah.
So the lytic switch protein, what's the name of it again?
RTA, also called OR50. Yes.
Do we know what turns that on? Because that's the sign to go into lytic phase, right?
Yes. And the RTA promoter looks like a human cell promoter. I mean, it looks very generic, and it has lots of transcription factor binding sites. And so, so the stimuli that turn on the RTA promoter, people have been studying elements responsive for that. So they've done some transcription factor mapping.
But we don't know what turns it on, right?
In real life,
it could be random, though, right? Just fires randomly, and then that cell goes into lytic, right?
Yes, it could be.
It's like sort of like lambda model, right? That's what we were
Right.
talking about at lunch today, I think. Okay.
We've learned a lot from lambda. Yeah. So.
You bet. Yeah. So there's a role for sensing RNA, which is unusual for a DNA virus.
Yes.
Right? But then the viral DNA, it goes into the nucleus, right? It's pretty much protected as it goes through the cytoplasm, goes in the nucleus. Yet you showed that DNA sensors in the cytoplasm, including CGAS and STING, which we've talked about on TWiV, these are sensing infection, right?
Yes.
So what are they sensing in the cytoplasm?
Yeah. So as with everything else in life, nothing is perfect.
No, RNA viruses are perfect.
So herpes viruses have a horrible PFU to infectious particle ratio.
Yes.
So I imagine so many different scenarios where the virus comes apart prematurely, it doesn't dock at the nuclear membrane, it actually comes apart in the cytoplasm, spilling out the viral genome. Another group, Soren Paludin, has shown that the capsids can be ubiquitinated and degraded in the cytoplasm, so that would release the DNA.
Yeah, right.
I also think like an infected cell that's been replicating bursts open, and you have all this DNA being spewed out, and that can be taken up by other cells in the area.
True. Yeah,
and so that's viral DNA in there too. So that could activate a cytoplasmic. So when I think of the possibilities of all of this happening, I think it's a pretty real life kind of scenario.
Can I move this? Because I'm getting nervous about your hand.
Okay. You've done so many of these, so you know exactly.
I know all the things that can happen.
Yeah.
And I still can't relax, though. That's the problem. So you think these leaky events, basically, or picking up DNA, but this is a whole sensing system that's in the cytoplasm. It seems to me that, well, I guess there are some viruses that do replicate in the cytoplasm, like pox viruses,
yes.
right? So maybe they're these evolved in part for them.
Yeah.
So then the virus, but then there are nuclear sensors as well, right?
Exactly.
You haven't looked at those, I think.
IFI-16, no. So another group, Balachandran's group, has looked at IFI-16 with KSHV previously.
And does it sense DNA in the nucleus,
yes.
right?
Yes.
So if you're not sensed in the cytoplasm, you're going to get sensed in the nucleus.
Exactly.
eventually. So again, in both cases, DNA binding to IFI-16 is going to turn on interferon production.
Exactly, yes.
And binding to C-gas and sting in the cytoplasm will
also
do the same thing,
exactly,
right?
yes.
The result is you get all these interferon-stimulated genes that inhibit virus replication. So why does the virus ever work at all? With
I
all
know,
this
isn't that amazing? So we think like probably that's why herpes viruses are so large. They have so many viral proteins that are built to evade these sensors or at least dampen them down. I don't think they can completely get rid of the censoring machinery, but they actually help to calm the sensing down a little bit. And you know, DNA in the cytoplasm is a trigger for sure. Everybody sort of understands, okay, I can see how, you know, you have DNA in the cytoplasm. That's just wrong. The cell needs to activate a sensing pathway for that. But DNA in the nucleus, it becomes a very catch-22 kind of scenario because you have host DNA, you have your home cell DNA, and you have the viral DNA. So how does nuclear sensing for DNA differ from virus to host? What are the differences? And that's very puzzling, and nobody has a good answer yet.
I thought you were going to answer it.
I wish I could.
So when the viral DNA gets in the nucleus, it's chromatinized?
Initially, it's not. So when it's just injected, it's not. But as soon as it is injected into the nucleus, then it does get chromatinized.
So it looks very much like host DNA.
Right, and then it circularizes. So I think the circularization also helps it to be stable because if it had open linear ends, that would be, a red herring for the sensing machinery. So I think, yes, I think it circularizes to prevent that from happening. But yeah, I mean, I don't understand how the nuclear sensors can distinguish between.
So that's still a big problem.
Big problem and big question, and anybody who answers that, my hat's off to them.
Because it does not sense. IFI-16 in the nucleus of an uninfected cell doesn't do anything, right?
Right.
It's not making interferon.
Right. And all the herpes viral DNAs, like the genes look like cellular genes, the promoters look like cellular promoters. It's not, there's nothing unique about it.
Not the circle itself, is it?
It could be. Maybe you've hit on the answer.
I mean, if you put a plasmid, when you transform plasmid DNAs into cells, they go in the nucleus,
yes.
right? Does that turn on IFI-16?
People have shown, I don't know about IFI-16, but it turns on C-gas and sting because they're moving through the cytoplasm. But yes, so it could be that, IFI-16, maybe Dave Knipe's lab has looked at this. Or maybe it was just a talk from his lab that I heard at herpes virus conference. But yeah, I think he's shown that there may be some sensing going on with IFI-16 as well during transfection.
So when it circularizes, it doesn't get supercoiled because it's not replicated, right? It just gets joined into a circle, and then it's this rolling circle replication, or else it's tethered to the... So when it's tethered, it's no longer sensed?
So that's a great question too, right? So, and nobody has shown either way.
So in a latently infected cell, where the viral DNA is attached by this Lana.
Yes.
I'll never forget it, because Lana, can't remember the singer's name. If I had my computer, I could say. Those cells, IFI-16 is not activated.
So what Balachandran's group, who showed IFI-16 recognizes KSHV genome, he was using latent cells, and he said that in latent cells, IFI-16 is activated, yes.
So they're making some interferon, but it doesn't matter because there's no virus replication, right? Is it a low level of interferon?
Yes.
And you think that might be actually needed?
So my hypothesis is that, yes, I think it's required for the life cycle.
So you said the genome encodes a lot of antagonists of interferon.
Yeah.
Otherwise it wouldn't. Every virus has to have at least one, I
Yes.
always say. And herpes viruses have many. And
Right.
you've discovered some of these, right?
Yes, yes. So we did a screen, at least for C-gas sting inhibition, and we found several viral proteins that are capable of inhibiting this pathway. So I think, and we don't know, we've worked out one of them, VRF1, but the other 5 we don't know the mechanism for, so we're still actively pursuing
So
that.
the IRF1 was published just in JV, right?
In PNAS, so there were 2 papers. So the C-gas sting screen was published in PNAS and had the VRF1 story. I have a second VRF1 story that just came out in JV as well,
Right.
where it antagonizes TLR signaling.
Okay, separate from, so that would be sensing RNA?
Yes,
It's
yes.
one of the
Yes.
TLRs that sense RNA?
Yes.
Okay. So the RF1 homolog, you designed a screen to look for inhibitors of C-gas, and that's
Yes.
what you pulled out of it,
Yes.
right? So how does that work? How does that inhibit C-gas?
Yeah, so we think it's not inhibiting C-gas per se, but it's inhibiting sting, which is downstream of
downstream of it,
C-gas.
right.
And interestingly, sting is now, was originally thought to be involved in only DNA sensing, but now papers are coming out saying that it's also involved in RNA sensing. So we think by targeting sting, it's sort of at the nexus of DNA and RNA sensing. And so the virus is chosen to attack a point in the pathway that's common. And so we showed that VRF1 can bind to sting and prevent activation of IRF3 and phosphorylation of IRF3, so thereby preventing interferon.
So do you think that inhibition of sting by this viral protein not only is blocking DNA but RNA sensing?
Yes, we think that that's definitely a possibility.
These RNAs that are in the
Yes.
tegument, so they may not just be sensed by Rig-I, they could also be sensed by, sting.
Yes.
It's sting that would do the sensing, not C-gas, right? Or
Right. Yeah. So and sting works through adapter proteins. Sting won't bind RNA directly, but it will work on bind RNA sensors
Okay.
that interact with it.
So the outcome of infection can be the cell responds with a great interferon and clears it.
Yes.
Which probably happens a lot.
Yes. Yes.
Or sometimes for reasons, random reasons, or maybe the virus makes a little bit more of the antagonist and it gets the upper hand and can prevail. Is that what you're thinking
yes.
might be happening?
And essentially you just need one virus to do that. So as you said, most of the time the cell is successful and eliminates the virus. But all you need is one, right, to establish latency. And then that can replicate and disseminate in the body over time. So I think, yes, so you don't need a lot of viruses to do that.
I'm looking at my cheat sheet here.
Oh, okay.
I've covered almost everything I wanted to talk about.
Yeah.
I have no idea how long we've been talking. I always lose time. So that's pretty cool. So there's basically a race between whether the cell or the virus wins. And, if you have this is the way I look at it. If you have all the right interferon pathway and sensor sequences, then
Yes.
it works. But if you have a polymorphism that makes one part of it a little...
Right.
So you're probably familiar with the work of Casanova,
Oh, my
right?
goodness, yeah. It's amazing.
Point mutation in a gene encoding a sensor can make the difference.
Yes, exactly.
And I think that's why the population has such a varied response to infections.
Yes.
We used to think everyone was pretty much the same with a few exceptions, right?
Yeah,
But
yeah,
everyone's
exactly.
different.
And where does he find these people with these cool mutations
well,
and TLRs and
If you notice his
STAT?
papers, he's got tons of authors, and I think a lot of those are involved in getting the patients. So you probably know there's polio vaccine associated paralysis.
Right, yes.
Right? Very rare event that happens. I think, it's 1 in 1.5 million recipients. I think that's a polymorphism of the same kind.
Oh, that's cool, yeah.
That these kids just have because innate immunity is so important in the early days of polio infection, I think that they have a polymorphism. They can't sense it properly or make interferon or whatever somewhere.
Right,
And it's just a matter of getting their genome
yeah,
sequences and doing it, because we know who has vaccine-associated. This is not very hard to find.
Yes,
They're there, the medical record. All you need is a little blood to do that. So that's always been my pet hypothesis. But who knows?
That's great. And also the immune system plays a role now, that the recent paper that came out showing.
Absolutely.
Yeah.
It's great. What we can do now is so cool. We can look at so much and do great sequencing, transcriptional analysis. I loved your experiments that you talk about where you You look at the transcriptome, all the ORFs and how they're transcribed, and you can do the, was that RNA-seq analysis?
That was just real-time PCR, but we can do the same with RNA-seq.
So you show a picture with like 100 viral genes, and you color each one depending on its level. So blue is in an infected cell, they're all pretty low. And then if you take away
C-gas or sting,
the sensors, which are keeping it down, then they get all red, they go way up.
I know,
it's so cool. Visually beautiful that you can see this, right?
Yeah.
And just think, you know, when you were studying
SP40
T antigen, if you could do this, right?
I know.
how much you could learn. So we can do so much nowadays. And what bothers me is that there's not enough money anymore. Right. People find it hard to keep working, and we're able to do so much.
Yes.
And new people are not coming in the field because they think it's not possible to work. That
is the sad part. But I have a confession to make, because all those viral genome profiles were done by Dirk Dittmer, who I'm married to, so I don't have to pay for that.
He never says no to doing a profile? No? Oh boy, that's great. My wife is a scientist, but she's not here. I don't collaborate with her, and if I try and talk about work, she doesn't want to hear it.
Oh, really?
I don't want to hear it.
She worked on picornus too, right?
No, she's not a virologist.
Oh, okay.
She's a pharmaceutical person.
But originally, as a grad student, I thought she was a virologist.
She did phage.
Oh, she did phage. Okay, well.
So we used to actually... We did talk back then, and I told her some experiments to do, and I had been doing sequencing, and I taught her how to do that. Yeah, so she got some interesting experiments. But now most recently, she actually works in where you live, Chapel Hill, and works on drugs for heart attacks.
Right. Oh, those are, big money.
That's a big deal, because a lot of people have heart attacks, but she doesn't want to hear about
So she does GPCR kinds of work, because most of the drugs for heart disease are GPCR drugs. So I'm curious if she's doing...
I don't know. They're nitrate-based compounds. This company has a proprietary chemistry that they're exploiting, but I'm not sure what the target is.
Okay, yeah.
She may never have told me. In fact...
Well, you can find out tonight when you talk to her.
I might, depends. But she certainly doesn't want to hear about viruses. I can't tell you on the
Really?
family show what she says when I start to talk about viruses.
But you've done so many podcasts, so she hasn't listened to any of them?
I don't think so. That's why I talk and I complain, and she never finds out because she doesn't listen to them. But I do want to do one with her at some point, because she has this wealth of pharmaceutical industry experience.
Yes.
I'd like to hear about, how things work and how you do drug screens and all the issues involved in developing drugs, which is what she's been doing. So hopefully she seemed like she would do that at some point. One other thing I wanted to ask, and then we'll stop. You have a translational title.
Yes.
What does that mean? What do you do?
Oh, so I have another hat in the Cancer Center, which I'm very passionate about. So Dirk and I both co-direct this program called Global Oncology. And it started out just looking at cancers in HIV-infected patients across the world. So here I'm talking about like developing countries, so India being one, Africa, South America. And so, but now we've, blossomed, I should say, to looking at cancers globally, not just viral cancers, but other cancers as well. And this all came about because both the World Health Organization and ACS, American Cancer Society, predicted by the year 2030 that 70% of the world's cancer burden is going to be in developing countries, because in Western Europe and in the US, we've gotten so good at diagnosing and preventing cancer before it actually becomes fulminant, that we'll see less and less cancer burden here, but the major burden will be in these developing countries, and they'll be hard hit, and it will have a lot global impact. So we've been working with different sites in different countries looking at viral cancers mostly, and then other people in our program are looking at non-viral cancers. So this includes KSHV cancers, but also Epstein-Barr virus, HPV, and this includes basic science as well as translational science. So like, how do we treat these cancers in a low income, resource limited setting? What kinds of chemotherapies are there that are cheap that can be used to treat these cancers? So we're involved in some of that, and that's been exciting to do that. We actually have now grants with places in Africa and India to do this.
Can you treat any of the KSHV-associated cancers?
Yes, so chemotherapy, so doxorubicin.
They're not 100% fatal.
They can be, but if you treat them at the early stage, you can
have
cure them.
remission,
Yes, yes. And they may come back later, but you can extend the person's lifespan.
Because you mentioned earlier, in Africa, 100% are seropositive for
Yes,
KSHV.
there's a high.
Wow, that means you're going to have a lot
Sub-Saharan, yes.
of sub-Saharan Africa.
Yeah.
So the virus, once you have it, that's it. You're stuck with it for
Right,
life.
right.
So
Unless we figure out a way to cure it. And that's why LANA, the protein that we talked about, is so important. If you can figure out how to get rid of latency, that would be a big deal for...
Could you just give people a microRNA to knock down LANA?
Yes, I guess you could. But you'd have to target every single cell that the virus is in, right? So that's sort of hard to do. And so it's the delivery is always where you get stuck at. Yeah.
And I mean, in a typical person, how many cells would be infected latently? Do you have any idea?
I have no idea.
It's not every
that's a
cell in
great
the body,
question. No, it's
obviously.
not every cell for sure. But it's, there are few cells, but they are spread out all over. So
So they're endothelial and B cells,
exactly.
right?
And endothelial cells go everywhere, right?
Yeah,
Because everything
right.
needs blood.
That's a horror drug, because most drugs, you have one place you have to get the
Right.
drug. So you get it in the blood and it goes to various organs. But most viral diseases, are not everywhere that you have to treat. Their liver
Yes.
or
Yeah. Yeah.
whatever. But that's tough when you have to get it into endothelium and then
B cells, which
as
also
well.
traverse all over.
Although, and it's not just B cells in the blood; they're in tissues,
Yes,
right?
yeah,
So that makes it
yeah.
harder. But are companies interested in developing therapeutics that would get rid of latently infected cells?
I think they would be if you had a good compound, because I think it would, if you could get a compound that worked, and you could also then apply it to EBV, which has a very similar mechanism of staying latent. I think they would be interested in developing that.
In the meantime, the approach is simply to, when you see a cancer, to try and
Yeah, we
cure
treat it with chemotherapy, like regular chemotherapy that you treat other cancers with. There's no viral-specific chemotherapy.
So the key is to catch it early.
Yes.
So if
yes.
it's not on the skin, how do you know?
That's
You
exactly
can't-
the problem.
So there's
yeah.
no test that you can do, a routine test that would detect a cancer at this
Not
point?
a routine one, but yeah, you could do like PET scans and things like that, but clearly that's not routine.
Yeah, right.
So,
And they would have to be a certain size already, right?
right. Oh, yeah. They also do like FDG PET, where the tumors tend to be more glycolytic, so you give them a little radioactive tracer, and then you can see your body light up at certain areas.
Okay. So what's your future? Are you going to continue to have a lab and do research, train people? Is that...
I think
you want to
so.
do that forever?
I think so.
Yeah?
I'm having fun doing it right now, so I don't see any reason why I should change that. So, yeah, I love training people and having a positive impact on my environment. So I'm happy doing what I'm doing.
So there's no question that science is in a rough patch now because
Yes.
there isn't money. I mean, what do you tell people who are students or postdocs about their prospects? Do you tell them the realistic facts, or do you try and not do that?
Yeah. So most of the people that I currently have want to work in industry. I think they're scared of academia and how competitive it is. But there are a couple of my students that want to do academia, and they're very driven. And I think the ones that really want to do it will do it, and I just support them in every way I can. But yeah, I sort of listen to what the person wants to do. I'm not one of those PIs that imposes my will on them and say, oh, you have to do this, you can't do
Sure.
that. So I try to be as supportive as I can.
I agree, but the problem is we don't know how to train people for other than academic
Yes.
or maybe industry. 'Cause
Yes.
we teach people how to do science, right?
Yeah.
But there are many other things you can do with
Yeah.
a science
Yeah.
degree. You can go into business or law, which means
Yeah.
you have to go to school, so that's fine. But if you want to write or communicate or do graphics, we
Right.
can't teach them.
Yes,
So I
yes.
feel in a way that we're limiting our trainees, that we don't teach them the whole picture.
And I think NIH has cottoned onto that. So at UNC, we now have this training grant. It's a very new training grant called the BEST training grant. And it's for training scientists in different areas. So just like you said, doing internships with science writers or in a company.
Right. That's really good.
And I think that's what we need. I'm of the opinion that we should have as many scientists as we can.
I agree.
But
I totally agree. Our world is the
Yes.
way it is because of science and technology, right?
yes, yes.
So how can you eschew it? It's scary. I'm reading some books by Isaac Asimov.
Oh,
Foundation. which is way in the future. And it goes through a period where the galaxy has abandoned science.
Oh, wow.
And you know, you're sitting here today, we have all this great science. You think that could never happen, but in thousands of years it could.
Yeah.
People lose track of what's important. They forget all the science and how to do it and so forth. So we have to prevent that.
Yeah, well, look at the anti-vaxxers. I mean,
exactly,
they've never seen a polio-ridden child or a small-pox-ridden person. Like, they have no idea.
Anti-vaxxers, anti-GMO, there's so much anti-science. It
Yes.
really bothers me because it's such a great thing. I mean, we're not claiming we have answers to everything, but if you give us time and make more scientists, let them investigate everything, we'll figure things out, you know. One of the things I think we don't train our students in is computational science, which I think you need a lot for biology.
Nowadays, yes.
And I think if everyone came in and had a good training, then they might say, well, you know, I want to do this for a career and go off and do that, and that would really help as well. Well, I think it's time to wrap this up and go have some dinner.
Yes, sounds great.
Is that a good idea?
And wine.
Oh, great. I'm happy to do that. And you have to just walk to your hotel, so you don't have to worry about that. So this episode is like all the others. You can find it on iTunes. You can find it at TWiV.tv. And if you have questions or comments for Blossom, we didn't talk about your name.
I thought you were going to ask me about my name.
Oh, so you got that when you were born, right?
Yes, I was born in the 60s, so I'm a child of the 60s. And
do you know whether it was your mother or father who picked Blossom?
My mother.
She picked Blossom?
Yes, yes.
And I remember when I first met you at a study section, I saw your name and I said, oh my gosh, I've never met a Blossom before. And then you had a good attitude, you were funny and you were friendly. So I said, it goes perfectly with the name. So does going through life with Blossom never bothered you? Or
well, I did get picked on a lot, as you can imagine.
In school, right?
Oh, yes. Oh, yes. But then I learned of Blossom Dearie, who's a jazz singer here in New York City.
That's true. That's right.
And so I figured,
there are other Blossoms out there.
Yeah, and I always figured it could be much worse. I could have had a name like Apple or China or Moon Unit.
Vincent.
Yeah.
Moon Unit, that's right.
Yes,
that's right.
Frank Zappa's.
Frank Zappa's kid.
Yeah,
so you're a 60s child, yeah.
Yeah,
That's cool.
So I prefer Blossom to Moon Unit, for sure.
No, Blossom's a great name. It's very unique, for sure. So if you have questions for Blossom, send them to TWiV at TWiV.tv. And this episode, as I said, will be found at TWiV.tv and also on iTunes, where you can subscribe for free. So I want to thank you for joining me. You're a professor at University of North Carolina, Chapel Hill. Many thanks for coming, not only to give a seminar, but for doing a TWiV.
It's been wonderful. Nice chatting with you about science.
Did you like this TWiV experience? It wasn't so bad.
Yeah, no, it was great.
It's just sitting around and talking. It just happens to be recorded, which to some people is inhibitory, but you get over it after a while.
Exactly,
you forget about it. You just talk, because I love to talk viruses.
Yeah, and it's so fascinating. There are all these different viruses. They all do things differently. Some of them do it similarly, but a lot of them just do it.
It's so cool, yeah. And this was the first TWiV in front of the wall of polio. This is the version 3.
So
this is
you glued.
I glued
You
them.
glued each. I read that on your blog.
Yeah, because the ones in the lab we just stacked and they would fall periodically.
Okay.
And this one I started at the bottom and I used crazy glue. I went through like 4 little bottles of crazy glue. And I went around the switch and the thermostat. And now I'm about a foot from the ceiling.
Yes.
And I do want to fill it.
You do want to fill it?
But I can't reach it.
You need a ladder.
I need to get something to get me. Because I have a few more plates. These are 6 well plates that we do.
Okay.
And they're all plaque assays.
And HeLa cells?
HeLa cells. Every one of
awesome.
these is a HeLa cell. I have to tell you a secret. It's not just polio. There's a few rhinoviruses in there as well. But I don't know which one.
That's great.
I can see here the plaques.
Yeah.
Anyway, I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.